My name is Loganjnelsen.
My name is Susana Melgar, and our research project explored the effects of tryptophan supplementation on anxiety and learning. We decided to explore this topic to research potential treatments for individuals who suffer from anxiety disorders. Because the literature shows that anxiety negatively affects learning and cognition, we were particularly interested in the effects during adolescence when humans are involved in formal learning. Previous research has shown that increasing tryptophan, an amino acid and precursor to serotonin, decreases anxiety-like behaviors in rodents and humans. However, studies have yet to examine the effects of tryptophan on learning. Thus, we predicted that acute dietary tryptophan supplementation would decrease anxiety-like behaviors and increase learning ability in mice. We used 29 adolescent male mice. The control diet consisted of 0.2% tryptophan, while the experimental diet consisted of 1.2% tryptophan. Both experimental and control diets were administered 24 hours before each behavioral test. We used the elevated plus MACE to quantify anxiety levels, and the object recognition task to measure learning ability in mice.
According to the literature, anxious mice prefer the closed arms of the elevated plus maze more than their less anxious counterparts do. As shown in Figure 3, tryptophan supplemented mice were significantly more anxious than the control group as they spent significantly more time in the closed arms. The object recognition task is depicted in Figure 2. For this measure, we first exposed a mouse to two identical objects for 10 minutes. After an hour, we reintroduced the mouse to the arena with one familiar and one novel object, and we recorded time spent near each object for 5 minutes. Less time spent near the familiar object and more time spent near the novel object indicates greater non-spatial memory. Figure 4 illustrates our finding that tryptophan-supplemented mice had worse memory than the control group. They spent significantly less time near the novel object and more time near the familiar object compared to the control group. In fact, unlike controls, the tryptophan diet group spent a similar amount of time near each object and thus did not indicate the ability to differentiate between familiar and novel objects. In conclusion, our data support the opposite of our hypothesis. Supplementing tryptophan in the diet increased anxiety and worsened memory. Further studies should focus on ideal dietary tryptophan concentration, timing of consumption, and how tryptophan affects gut microbiota, which might in turn affect the brain.